Hello, and a very warm welcome to everyone to our today's webinar on targeted therapies in lung cancer clinical trials, evaluating efficacy and overcoming resistance. I am Anvesha Bhattacharya and I represent Veeda Clinical Research. Veeda Clinical Research Limited, together with its subsidiary, Bioneeds India Private Limited, referred to as Veeda Group, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovator biosimilar and generic drug development programs for our global clientele. Together, we serve clients globally in pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal nutraceutical, device industries. As your facilitator, today I would like to introduce you all to a couple of points as we begin this session. This session will have a presentation in the beginning, followed by a Q&A round. We have enabled our Ask a Question feature. It's right on your screen where you can drop all your questions related to the webinar right there. Now, moving forward, I would like to introduce you all to our speaker and panelists for today's session. Dr. Ravi Alamchandani is a MD Pharmacology Gold Medalist with over 9 years of experience in field of medical affairs and safety reporting. His experience includes working in various hospital, pharmaceutical companies, and contract research organizations like DDMM Heart Institute, Torrent Pharmaceuticals, Lambda Therapeutic Research before joining Veeda Clinic. He is currently associated with Veeda Clinical Research and is leading the team involved in medical monitoring, medical writing, and pharmacovigilance. Dr. Ashutosh Jani, PhD, Pharmacology with over 17 years of experience in the field of clinical research. He has experience working in academia pharmaceutical companies and contract research organizations like Claris, Acutis Research, and Lambda Therapeutic Research before joining Veeda. During his tenure, he has managed teams involved in clinical operations and BD and is having good rich experience in clinical development, project management, contract negotiations, and implementation of programs in therapeutic areas like oncology, psychiatry, and endocrinology, including multiple rare and complex indications. Our third panelist, Mr. Sandeep Yergude, currently working as AGM in project management at Veeda, has expertise in sponsor relationship management, pre-study initiation, interim, and closeout monitoring. He is also involved in developing a project plan, including all elements listed in project template as appropriate for the project, and coordination in internal and external clinical development activities of all members involved in design, conduct, and assign clinical trial. So without any further delay, I would like to virtually call upon Dr. Ravi to begin this session.
Yeah, hello everyone. Next slide, please. We are here to discuss targeted therapies in lung cancer and the clinical trials of these therapies. Now, whenever we used to hear cancer, we used to hear chemotherapy. The next image that would be there in our mind would be patients with severe hair loss, patients with severe weakness, patients with oral ulcers, and so on. This is because the therapies, the oncotherapies that were there initially, the chemotherapies, they were non-specific and they targeted any cell that replicated at a particular rate or beyond that. Thus, these therapies, they not only killed cancer cells, but they also affected rapidly growing cells in our body, like those present on our skin, on our mucosa, and so on. With time, scientists identified the specific areas or specific targets or specific receptors or specific genes that are responsible for the rapid multiplication of these cancer cells. So targeting these receptors or these areas would specifically control or limit the growth of these cancer cells without affecting the normal or other healthy cells, and thereby reducing the overall toxicities of these therapies. Today, we discuss these therapies in terms of lung cancer. Lung cancer, why? because still it is the leading cause of cancer-related mortality worldwide in both men as well as women. Lung cancer in itself are divided into two broad categories: small cell lung cancer and non-small cell lung cancer. Of the two, the vast majority of lung cancers are non-small cell lung cancers, comprising around 85% of the lung malignancies. These lung cancers are divided based on their histological subtypes into squamous cell carcinoma, large cell carcinoma, and adenocarcinoma, with adenocarcinoma being the most common subtype of NSCLC. Surprisingly, 2 out of 3 advanced NSCLC patients, they have one or the other genetic alteration that include KRAS, BRAF, EGFR, MET, or ERBB2 mutations, or ALK, ROS1, RET, NTRK genome rearrangements. Of these, the most common genetic alterations seen in NSCLC patients are those related to epidermal growth factor receptor, which is seen in around 10 to 13% of patients. So we see that, specific mutations or genetic rearrangements, they result into lung cancer. So if a therapy is targeting to these particular rearrangements or mutations, then these therapies, they kill only cells harboring these mutations and thereby not affecting or minimally affecting other cells in the body. Next slide. So as we discussed a few minutes earlier, conventional chemotherapy used in the treatment of lung cancer not only kills the cancer cells, but also affects the normal cells, resulting into untowards or unwanted side effects. Newer therapies are targeted and they specifically interrupt the growth of cancer cells and thereby help reduce the damage to the healthy cells. Targeted therapies can be used for management of cancers that have specific genetic abnormalities in their tumor cells. These specific genetic abnormalities can be detected through biomarker testing that looks for specific changes in the tumor cells, DNA, such as mutation, addition, deletions, or rearrangement. So basically, there are various tests available these days wherein you can identify the specific genetic abnormality seen in a cancer, and then you can specifically target those anomalies and manage the cancer. Newer treatment modalities can target these changes directly and cause fewer side effects, as they focus on targeting cancer cells that have specific genetic alteration and thereby they spare the normal cells. The specific target may be on the surface of the cells or maybe inside the tumor cells. Next. So the currently approved therapies for lung cancer can be categorized as EGFR inhibitors, ALK inhibitors, ROS1 inhibitors, BRAF inhibitors, and MET inhibitors. The recent administration of TKIs as first-line regime for ALK rearranged and EGFR mutated NSCLC has shown to outperform the platinum-based chemotherapy in terms of response rate, in terms of progression-free survival, and in terms of overall survival. So we reduce these therapies, they not only decrease the side effects, but they also have shown to have better survival or better progression-free survival outcome for the patients having lung cancer. Next. Now we discuss few of the therapies. The first one to be discussed is epidermal growth factor receptor and its inhibitors. EGFR is a transmembrane protein that is involved in signal transduction pathways. It is critical in regulation of cellular proliferation and survival. It is normally expressed in many different cells, including epithelial cells. In some neoplasms, there is overexpression and dysregulation of EGFR. Activation of tumor cell EGFR through autophosphorylation triggers a series of intracellular events that include cellular proliferation, blockage of apoptosis, invasion and metastasis, and tumor-induced neovascularization, all of which are hallmarks of cancer. There are two categories of drugs that affect EGFR: the monoclonal antibodies and the tyrosine kinase inhibitors. The monoclonal antibodies that are available to target EGFR are pentimumab and cetuximab, which are administered intravenously. They act through extracellular binding and subsequent inhibition of EGFR signaling pathways. Then we also have tyrosine kinase inhibitors like erlotinib, gefitinib, lapatinib, which are administered orally. They act by intracellular binding and subsequent inhibition of EGFR signaling pathways. So these therapies are available. These therapies are easily being made available to the patients, wherein they have shown to improve efficacy. And the common side effects that can be encountered with these therapies are skin rash, dry skin, stomatitis, diarrhea, and sometimes loss of appetite. Next. So here we see how EGFR hyperactivation can result into, cancer generation, because these cells, they directly affect cellular growth. Being mutated, they result into uncontrolled growth and thereby cancer. Next. Another receptor that has been identified to result in cancer when found to be mutated is ALK, or anaplastic lymphoma kinase. ALK gene provides instruction for producing ALK, a protein that helps regulate cell growth and division. It plays a pivotal role in cellular communication and in normal development and function of nervous system. When mutated, it causes abnormal production of ALK, which then results into uncontrolled growth and spread of these cancerous cells into the body. According to American Cancer Society, around 5% of NSCLCs have alteration in ALK gene, and these are labeled as ALK positive NSCLCs. ALK is a potent oncogenic driver in lung cancer. ALK tyrosine kinase inhibitors, they result in significant benefit in patients with ALK positive lung cancers. Next. The various ALK therapies that are available currently are crizotinib, cabozantinib, certinib, enectinib, alectinib, and so on. These are oral medications that bind to abnormal ALK protein, block the protein from energy sources, and deactivate it. They help in the shrinkage of the tumor and slow the growth of the tumor, and thereby improve the survival chances of the patient. Next. The common side effects that can be seen with ALK inhibitors are nausea, vomiting, diarrhea, constipation, fatigue, and vision changes. Less, but some of the severe side effects seen are inflammation in lungs or other organs, liver damage, nerve damage, abnormal heart rhythms. Next. ROS1 inhibitor. So ROS proto-oncogene 1 encoded by ROS1 gene is located on chromosome 6q21.2. It belongs to the subfamily of tyrosine kinase insulin receptors. It is one of the 52 receptor tyrosine kinases and one of the 2 orphan receptor tyrosine kinase where its ligand is still unknown. It is evolutionarily related to ALK. ROS1 rearrangement was discovered in glioblastoma in 1987 and in NSCLC back in 2007 and in cholangiocarcinoma in 2011. It is a validated therapeutic target in NSCLC and accounts for 1 to 2% of all the lung cancer diagnosed. Different techniques are employed to identify ROS1 rearrangement like immunohistochemistry, FISH technique, RT-PCR, and so on. There are currently 2 FDA-approved options for managing these ROS1 mutations, crizotinib and entrectinib, which are administered orally. These also act as ALK inhibitors, these 2 drugs. The common side effects include dizziness, diarrhea, nausea, vomiting, and hepatotoxicity. So we can see that some of the TKIs, they not only inhibit ROS1, but they also inhibit ALK, they also inhibit EGFR. So these tyrosine kinases, the family of tyrosine kinases, they are inhibited all together or some of them together by these newer drugs called tyrosine kinase inhibitors. And thereby, if there is some cancer that harbors more than one mutation, even these can be targeted and managed with these tyrosine kinases that are available today. Next. BRAF inhibitor. BRAF is VRAF murine sarcoma viral oncogene homologue B, which is a member of RAS kinase family. A member of the RAS kinase family is a proto-oncogene and plays a critical role in cellular growth, proliferation, and differentiation through mitogen-activated protein kinase pathway. So basically, these inhibitors or these genes under normal circumstances, they tend to prevent cancer by signaling, by generating signals whenever the abnormal cells are developed. So you have an abnormal cells, these receptors get activated, they signal to the body that there is an abnormal cell and they help kill it. But what happens when these genes or these receptors are mutated in themselves? They will not control the growth of these abnormal cells, and thereby these cancer cells, they just go on multiplying, mutating, and spreading. Incidence of BRAF mutation in NSCLC is low and accounts for 0 to 3% of all the lung cancers. Vemurafenib is a potent inhibitor of BRAF mutant family. Vemurafenib is derived from its ability to inhibit V600E mutated BRAF. Dabrafenib is a potent ATP competitive inhibitor of BRAF kinase and is highly selective for mutant BRAF A kinase panel screening, cell lines, and xenograft. Then there is tremetinib, which is a type 3 reversible allosteric non-ATP competitive inhibitor of both MAC1 and MAC2 inhibitors, MAC1 and MAC2 receptors. Then there is oncorafenib, which is a newer generation BRAF1 inhibitor, which targets a key enzyme in the MAPK signaling pathway. Next. Then there is a MAT inhibitor, which is a mesenchymal epithelial transition, which belongs to a family of, again, tyrosine kinase. MET overexpression due to increased gene copy number or transcriptional regulation is commonly detected in many different solid tumors, including lung cancer, and correlates with poor prognosis. MET inhibitors may be non-selective, like mercitinib, gilasitinib, or may be selective, like capmatinib, tepotinib, tivantinib, and savolitinib. They are administered orally, and significant side effects include hepatotoxicity, dyspnea, pancreatitis. Other side effects like peripheral edema, dizziness may occur. Some patients may also experience musculoskeletal pain. Next. So we have discussed various targeted therapies. We have discussed various targets which can be attempted to be inhibited in case of patients with lung cancer when they harbor those specific targets. These genes or receptors are normally present in every individual. They are there to check the growth of the cancer. But when they in themselves become abnormal, they do not, or they are no more capable to inhibit the growth or multiplication of the abnormal cell, thereby resulting into cancers. So when you inhibit them, when you inhibit such cells, you help control the cancer cells from multiplying and thereby spreading, and thereby improve the overall survival of the patient. With this in mind, I'll pass on the further presentation to my colleague, Dr. Ashutosh Jani, to take us forward through our journey. Thank you.
Thank you, Dr. Ravi. I would like to take it up from here. So, as we know, like say lung cancer, non-small cell lung cancer is one of the predominant therapeutic indication as far as lung cancers are concerned. So, can you go to the next slide? Can you go to the next slide? Yeah. So, as you just seeing through that, non-small cell lung cancer is one of the predominant lung cancer prevalence. And what we've seen through is, like, say, if we globally chart the average recruitment rates for any typical non-small cell lung cancer type of a study, that varies from geography to geography, where we could see we just try to plot across different geographies where the recruitment rate is as low as a 0.02 and goes up to 1.68. So this is the type of heat chart where we try to plug in all the various recruitment rates across the world. Go to the next slide. If we predominantly focus on a European region, there also the recruitment rates have been quite varied, ranging right from 0.16 to 1.4 patients per site per month. The next slide. Our foray is India, where we have worked with various clinical trial sites across India. And our experience says that when it comes to India, the normal recruitment rate for any typical non-small cell lung cancer is around 0.3. And we also seen through that there are average dropout rates depending upon the different type of studies is as high as 77% dropout from the study. You can go to the next slide. So what we do at Veeda is we provide the sponsor a single point of contact for the study, who would be the project manager, who would be facilitating the total study conduct at Veeda. The project manager would be provided with ample support in form of CRAs, CTAs, medical monitors, regulatory department, as well as medical writing would be supporting data management and biostats. There will be a strong management oversight on the project manager and the project through the senior management team. So, as a project manager, the person would be responsible and accountable for the total project delivery. That includes the development of various plans, managing the study, the quality and on-time deliverables with respect to the study. So, the project manager would be a point of contact responsible for strategizing the study and meeting the expectations of the sponsor. You can go to the next slide. So we at Veeda have this accelerated startup group. So the typical process as far as any clinical trial application is concerned is what we try to do is we try to engage our sites on an early stage itself, which would then transcend into getting in easier and rapid easy approvals. Helps in streamlining the typical processes across the sites, and through which we target fast track recruitment. And what we've done is like a typical startup activity timelines is ranging nearly about 6 weeks of startup, right from the protocol to the easy approvals in place. We can go to the next slide. So as far as non-small cell lung cancer is concerned, we work with various sites, and we try to chart down a few of the sites who've been quite high enrolling sites as far as non-small cell lung cancer is concerned. So one of the topmost site is Tata Memorial Hospital, PGMER. We also work with various regional cancer centers across the country, which have provided an ample scope for recruiting patients of non-small cell lung cancer. To the next one. So we, with the regional experience and we understand the nuances associated with oncology trials, we at Veeda have that knowledge and experience of working with these proposed site, and provide a highly experienced team and effective project management on the project. So we've been more into training, training our resources that includes the CRAs, the site team, and there is a joint interaction with the sites and the project manager to ensure that we meet the recruitment timelines in consultation with the sponsor. Yeah. We can go to the next slide. So, as a part of our proactive approach as far as oncology trials, especially lung cancer trials are concerned, We ensure that there is proper and thorough training which has been given to the investigators and the site personnel, especially during the site initiation visit. We also ensure that there is a robust sample and other logistic transfers and associated with the study. We make a mock run from the site to ensure that there is a safe delivery of the samples to the sample lab or Veeda, whatever the case may be. We also ensure that the site infrastructure is robust. There are various equipments required based on the study demand, and they have the required capabilities to upload the images because these type of studies demand high network, strong network for uploading of sample images. The shipment to the sites are also ensured. So the shipment of the IMPs, we make mock runs, trial runs to ensure that the transit time between the shipment from Veeda to the site is within the expected timelines. We also ensure that the site has all the equipments which have been calibrated. That includes equipment as small as a digital clock or a weighing scale for that matter. So we provide these type of support to the staff site to ensure the requisite instruments have been available at the site. Go to the next slide. So with this, I would hand it over to our project manager, Sandeep today. So he will take us through the different nitty-gritties associated with the operational aspect of clinical trials on the lung cancer. Over to you, Sandeep.
Thank you, Dr. Astush, for driving us through the PPTs. So I will take up here from looking at the challenges what we are facing in the clinical trials. So can you go to the next slide? Yeah, next please. So when we conduct the trial, there are certain challenges and considerations which we need to take care. So these include the site selection, then patient selection, the biomarker selection, and product tracking in terms of the lab. So when we are selecting the site need to be have. Can you go to the next slide, please? Yeah, while site selection, the main factor which includes these are the experience and clinical trained site, the site which are having sufficient experience on the monitoring and these who can support the CRO for the monitoring, then patient coordination and the site accreditation in terms of the local lab accreditation for NABL. These are the parameters of the site selection. And next slide, please. So basically, we focus here on the levels of barrier in conducting clinical trials. So the barrier can be at any level, which includes patient level, then healthcare provider level, clinical level, institutional and structural level. Please, next slide, please. When it comes to the patient level, the trust that is most important factor while going into the clinical trial. So that's why the investigator need to convince the patient. They need to make him understand what are the advantage of the clinical trial, how it benefit. That includes. Then in terms of any SAE, what are the health coverage for the in terms of any SAE, then language, then immigration we need to check in terms like patient is traveling from one country to another country and if he want to settle in that country and he don't any the identity card of that country, that may also hinder the patient participation. Then potential quality of care that may receive from the trial from the investigator, that also is important. Most of the times the trial which we conducted, they are randomized clinical trial. So in randomized clinical trial, the patient is not aware what type of treatment he or she may get. So there is some doubt in his mind what type of treatment he might be giving, whether placebo, whether active treatment. So that may also one of the factor to lose the trust in the clinical trial. Sometimes the patient's distance from the clinical site, which may hinder his participation in the trial due to the long distance. So whenever we are trying to convince the patient, these are the factors which we need to take care so that we can increase his retention period in the study. Next slide, please. So the next level of barrier, which itself is the investigator. So if in any trial, the investigator is the prime importance because he is the person who is driving the clinical trial. He's the person who is well aware of the protocol, its inclusion, exclusion criteria, what are the benefits to the patient. So whenever trial is being conducted, the investigator should be thoroughly knowledgeable about the protocol. So that can be anything, it design, anything. So sometimes the time is very important factor for the investigator because they are so much occupied in their routine practice. So the time constraint can be a limited factor for the investigator. Then sometimes limited awareness about the clinical trial. And if they are aware, then non-support from the team, that can also the limit to the investigator. Then lack of awareness of the clinical trial is an important barrier to the patient because if the patient, the investigator is not aware about the clinical trial, so he cannot pass the information fluently to the patient so that they can be a part of the study. And as we discussed, the non-cooperation from the supporting department can lose the interest of the investigator in the clinical trial. Next slide, please. So the next level of barrier include the clinical trial itself, the design of the clinical trial. The complex the clinical trial or the study design, this can also limit the patient participation or investigator interest in the clinical trial. Sometimes extended follow-up in the clinical trial would delay the enrollment because longer the duration, patient may lose their interest to continue the study. Then the complex design, which have some restricted criteria to participate the patient, that can hinder the clinical trial. Next slide, please. Now the level of barrier which includes structural or institutional. This is the place where we are conducting the clinical trial. So the clinical trials are revenue based. So sometimes the institutional, they have constraint to distribute the amount to the investigator, which can slows down the clinical trial in that particular institution. Then oncology provides the routinely encounter bureaucratic barriers to implementing the clinical trials. Without dedicated staff, clinical trial cannot be conducted because if you see the clinical trial, it's all about the support function from the staff, from the PI, from the COI, or anyone from the person, those who are the part of the trial. Then patient enrollment, then follow-up, and limited support to the participant patient from the institution. And sometimes the delay in the IRB itself can delay the clinical program. So these are the various levels of barrier that can limit the clinical trial. Next slide, please. Yeah. So when we come to the protocol, the most important part is the eligibility of the patient. So this includes various factors that can allow or remove the patient from the trial. So while designing the clinical trial, eligibility criteria is very important. So we have to validate that inclusion exclusion criteria with the medical monitor before entering the patient into the clinical trial so that no wrong patient should be part of the study and that can cause or damage the entire clinical trial. Then patient condition, his comorbid condition, his ECOG status should also be taken into care while he's participating in the clinical trial. Then what are the concurrent therapy or supportive therapy in addition to the existing clinical trial that can be provided to the patient as a support function. That is also important part of the clinical trial to allow the patient in the study. Next slide, please. Yeah, when we are doing the trial, the sample collection is important factor because through the sample we came to know what is the biochemical parameter to that patient, so that on that basis patient can be part of the study, and it is good for the investigator to decide whether patient will be part of the trial or not. So the logistic issue is the main factor. Not sometime, but we are using the central lab in the study, and the sites are locate geographically. So we need to take care. The sample should reach to the central lab within a certain duration, because otherwise it will cause the sample deterioration and give the false result in the study. So these are the factors while sample collection and sample tracking. Also, the site should be keen, or they should be experienced on sample collection, so that no sample should be damaged. Yeah, next slide, please. Yeah, this was from my side. Any question?
Thank you. Now, moving on, we will be proceeding ahead with our discussion, where we are going to answer some of the interesting questions posed by our audience. So, my first question to you, Sandeep, is why experienced team is preferred in clinical trials and its operations?
Yeah, strong background in the therapeutic area, disease indication, and target patient population will help the site staff to speed up more quickly on the study design, recruitment strategies, and data collection and analysis tool to be used. So site having the previous experience with the disease indication and its associated standard of care in the countries where the study may be conducted is also extremely beneficial. And existing treatment patterns and guidelines for the disease of the interest may vary widely, and the specialist and site type may also differ as well. So this factor can have significant impact site and investigator selection, as well the potential for the patient recruitment. So working with the study team that has already experienced or familiar with the trial or with the indication or with the how to handle the patient, that can help the drug developer to expedite the clinical trial development program. So that's why the trial experience is very much preferred in the clinical trial.
All right. Thank you, Sandeep. My next question to you is why careful selection of patient is important in clinical trials?
Yeah, the clinical trials always run on the few factors. What is the inclusion criteria? What is the exclusion criteria? So based upon that, the patient may be enrolled in the study. So recruiting the planned sample size within the defined time frame in the clinical trial has proven to be the chief bottleneck in the drug development process. It may causes the missed clinical trial timeline, lead to the increased cost, and consume the more time and may other aspect of the clinical trial. So apart from recruitment of the participant, retention is equally important. So if we carefully select these criteria, then we can run the clinical trial very smoothly. As you see in the lung cancer, especially in India, the recruitment is very, very less, 0.3 around. So if we focus more clearly, closely on the inclusion exclusion factor, we can certainly come over such type of issues in the clinical trial. So that's why it's very important to have the careful selection of the patient in the clinical trial.
All right. Thank you, Sandeep. Thank you, Dr. Ashutosh. Thank you, Dr. Ravi.
Yes.
So I would like to thank all the panelists for nicely answering most of our audience questions. I would like to take a pause over here. We understand that this session has kept us all engaged throughout, but due to time constraints, we were not able to take any further questions at this point of time. But please be rest assured that we will be sending answers to every questions of yours via emails. Also, we will be sending across recorded link of this webinar on all registered email IDs. Lastly, on behalf of Veeda Clinical Research, I would like to sincerely thank you all for being here today and taking out the time to be a part of this webinar. I would also like to extend our gratitude to all the panelists for being such a wonderful presenters and giving all great learnings today. We at Veeda Clinical Research earnestly look forward to hosting you all again. Till then, take care and stay safe, everyone. Thank you so much.
Thank you.